Welcome to the first lecture on general pharmacology by Dr. Priya Sahay from GNP Sir classes. Today, we will explore the fascinating world of pharmacology, the science of drugs and their effects on the human body. Pharmacology is the science of drugs, derived from the Greek words, pharmakon, meaning drug and logos, meaning discourse. It involves the study of chemical substances that produce biological effects. We will delve into pharmacokinetics, pharmacodynamics, and the therapeutic uses of drugs. Pharmacology branches into several sub-disciplines, including pharmacokinetics, chemotherapy, toxicology, and more. Each branch focuses on different aspects of drug action, from genetic influences to the economic impact of drug therapy. Clinical trials are essential for testing new drugs. They involve different phases, starting with healthy volunteers and progressing to diseased patients. These trials help determine the safety, efficacy, and optimal dosage of new medications. Key terms in pharmacology include dose, dosage form, and dosage regimen. Understanding these terms is crucial for administering drugs correctly and ensuring their effectiveness in treating patients. Drug information sources like pharmacopoeias provide official information on drug properties, purity, and potency. These sources are essential for healthcare professionals to ensure the safe and effective use of medications. Formularies list drugs in both generic and brand names, promoting rational prescribing and cost-effectiveness. Other sources like Martindale and the Merck Index also provide valuable drug information. The history of pharmacology dates back to ancient civilizations like India and Egypt. Early texts and practitioners laid the foundation for modern medicine, with contributions from figures like Charak, Sushruta, and Hippocrates. Notable figures in pharmacology include Hippocrates, Aristotle, and Paracelsus, who made significant contributions to the field. Their work has shaped the understanding and practice of medicine over centuries. Drugs can be sourced from both natural and synthetic origins. Natural sources include plants, animals, microorganisms, and minerals, while synthetic drugs are created in laboratories. Medicinal plants have been used for centuries for their therapeutic properties. Different parts of plants, such as roots, leaves, and flowers, contain active compounds that can treat various ailments. Animals provide valuable sources of hormones, vitamins, and other therapeutic substances. These animal-derived products play a crucial role in modern medicine. Microorganisms like fungi and bacteria are sources of antibiotics and vaccines. These microbial products have revolutionized the treatment of infectious diseases. Minerals are another important source of drugs, providing compounds like antacids, purgatives, and diuretics. These mineral-based drugs are essential in treating various medical conditions. Synthetic drugs are created in laboratories and make up a significant portion of modern medications. Semi-synthetic drugs are modified natural compounds, while gene therapy represents a cutting-edge approach to treatment. Active principles of medicinal plants include alkaloids, glycosides, and oils. These compounds have specific pharmacological actions and are used in various therapeutic applications. Glycosides are compounds containing sugar molecules and have important pharmacological actions. They are used in medications for their therapeutic benefits. Oils, both fixed and volatile, have various medicinal properties. Fixed oils are used as purgatives, while volatile oils have applications in aromatherapy and other treatments. Resins, oleoresins, and balsams are plant-derived substances with pharmacological actions. They are used in treatments for their antiseptic, astringent, and other therapeutic properties. Gums and tannins are plant products with medicinal uses. Gums act as emulsifiers, while tannins have astringent properties and are used in treating various conditions. Drugs can be administered through various routes, including oral, sublingual, rectal, and parenteral. The choice of route depends on the drug's properties and the desired effect. Several factors influence the choice of drug administration route, such as the drug's physical and chemical properties, the site of action, and the patient's condition. These factors ensure the drug's effectiveness and safety. Local routes of drug administration include applications to the skin and mucous membranes. These routes provide targeted treatment with minimal systemic effects. Systemic routes are designed to distribute medication throughout the body via the bloodstream. The oral route is the most common and convenient, but it has limitations such as slower action and potential for nausea. Some drugs are not suitable for oral administration due to destruction by stomach acids. Sublingual and buccal routes allow for rapid absorption of lipid-soluble drugs, bypassing the liver. Rectal routes are used for irritating drugs, while the cutaneous route is for slow, prolonged absorption. Inhalational routes provide rapid absorption through the lungs. Nasal routes bypass the liver and rapidly absorb drugs through mucous membranes. Parenteral routes, including intravenous and intramuscular injections, offer direct delivery into the bloodstream or tissues, requiring sterile preparations. Intravenous routes provide immediate drug effects, while intradermal routes are used for small doses. Intraperitoneal routes are important for large animal practices. Various routes of administration offer different benefits and are chosen based on the drug and patient needs. Pharmacokinetics studies the movement of drugs within the body, focusing on absorption, distribution, metabolism, and excretion. These processes determine the onset, intensity, and duration of drug action. The process of drug absorption involves the drug entering the bloodstream from its site of administration. This diagram illustrates the journey from administration to excretion, highlighting key stages like absorption, metabolism, and excretion. Biological membranes, such as the plasma membrane, regulate the movement of substances into and out of cells. These membranes are composed of a lipid bilayer with embedded proteins, allowing selective permeability. Drugs can be absorbed through biological membranes via passive diffusion, facilitated diffusion, active transport, and pinocytosis. Passive diffusion depends on the drug's lipophilic-hydrophilic balance and concentration gradient. The passive diffusion of drug molecules across biological membranes is best described by Fick's first law of diffusion, which considers factors like concentration gradient and membrane thickness. Filtration allows drugs to pass through aqueous channels in membranes, driven by hydrostatic or osmotic pressure. Specialized transport involves carrier proteins that help drugs cross membranes, often against concentration gradients. Active transport moves drugs against concentration gradients using energy, while facilitated diffusion does not require energy. Endocytosis and pinocytosis involve engulfing particles or droplets for absorption. This table compares simple diffusion and active transport, highlighting differences in energy requirements, carrier involvement, and occurrence in the body. The pH partition hypothesis explains how the ionization of drugs affects their absorption. The Henderson-Hasselbalch equation helps calculate the ratio of ionized to unionized drug at a given pH. Exceptions to the pH partition hypothesis include drugs that are always ionized or unionized regardless of pH. These exceptions affect how drugs are absorbed and distributed in the body. The general rule for pH and pKa consideration is that acidic drugs ionize more in alkaline environments, while basic drugs ionize more in acidic environments. This affects drug absorption and distribution. Ion trapping occurs when drugs accumulate on one side of a membrane due to pH differences. Bioavailability measures the fraction of administered drug that reaches systemic circulation, while bioequivalence compares the bioavailability of different drug formulations. Drug distribution involves the reversible transfer of drugs between blood and tissues. Factors like blood flow, capillary permeability, and tissue binding affect how drugs are distributed throughout the body. Capillary permeability varies in different tissues, affecting drug distribution. Highly lipid-soluble drugs can easily enter the central nervous system, while ionized drugs cannot pass through the blood-brain barrier. Drugs bind to plasma proteins and tissues, affecting their distribution and action. Albumin is a major binding protein, and the degree of binding influences the drug's availability and effect. The clinical significance of drug binding includes lower volumes of distribution and temporary storage of drugs. Drugs can accumulate in specific tissues, affecting their action and potential toxicity. Lipophilicity affects drug distribution, with highly lipophilic drugs crossing biological membranes more easily. Redistribution can terminate drug action as drugs move from highly perfused to less perfused tissues. Drug metabolism, also known as biotransformation, occurs primarily in the liver but also in other tissues like the blood plasma, gut, and kidneys. This process transforms lipophilic drugs into more polar, hydrophilic forms, making them easier to excrete. Biotransformation is divided into two phases, phase 1 and phase 2. Phase 1 reactions often produce active or inactive metabolites, while phase 2 reactions generally result in inactive metabolites. Phase 1 reactions, also known as non-synthetic reactions, are primarily carried out by a group of enzymes called monooxygenases in the liver, which involve cytochrome P450. These reactions include oxidation, reduction, and hydrolysis, and they often introduce or expose functional groups on the drug molecule. Phase 1 reactions can activate or inactivate drugs and are crucial for drug metabolism. Phase 2 reactions, also known as synthetic or conjugation reactions, involve the addition of a chemical group to the drug or its phase 1 metabolite. These reactions usually occur in the liver and involve enzymes like glucuronol transferase. Phase 2 reactions increase the water solubility of the drug, facilitating its excretion in bile or urine. These reactions are essential for detoxifying and eliminating drugs from the body. Excretion of metabolites is the process of eliminating drugs from the body, either unchanged or as metabolites. The kidneys play a crucial role in this process, with renal excretion involving glomerular filtration, active tubular secretion, and passive tubular reabsorption. Efficient excretion depends on the drug's polarity, with polar compounds being more easily eliminated. Biliary and fecal excretion is another important route for drug elimination, especially for larger molecules. Drugs excreted in bile can undergo enterohepatic circulation, where they are reabsorbed and returned to the liver. Other routes of excretion include sweat, saliva, and milk, though these are less significant. Understanding these pathways is essential for comprehending drug elimination. The kinetics of drug elimination involve three fundamental parameters: bioavailability, volume of distribution, and clearance. Clearance is the rate at which a drug is removed from the plasma and is crucial for determining the drug's dosing regimen. Understanding these parameters helps in predicting how a drug behaves in the body and its overall efficacy. Plasma half-life is the time required for the plasma concentration of a drug to reduce to half its original value. This parameter is influenced by the drug's clearance and volume of distribution. First-order kinetics means the half-life remains constant, while zero-order kinetics means the half-life increases with time. Knowing the half-life is essential for determining dosing intervals. Prolonging the action of a drug can be achieved through various methods, such as sustained-release formulations, increasing plasma protein binding, and inhibiting metabolism or renal excretion. These strategies help maintain therapeutic drug levels in the body for a longer duration, enhancing efficacy and patient compliance. Pharmacodynamics studies the physiological and biochemical effects of drugs and their mechanisms of action. It examines how drugs interact with their targets in the body, such as receptors, enzymes, and ion channels. Understanding pharmacodynamics is crucial for developing effective and safe medications. Drugs can have various effects on specialized cells, including stimulation, depression, irritation, replacement, and cytotoxic action. These effects depend on the drug's mechanism of action and its interaction with specific cellular targets. Understanding these effects helps in predicting drug responses and potential side effects. The basic mechanisms of drug action include non-cellular mechanisms, chemical reactions, physiochemical mechanisms, and modification of body fluid composition. These mechanisms explain how drugs produce their effects and interact with the body at a molecular level. Understanding these mechanisms is essential for drug development and therapeutic application. Cellular mechanisms of drug action involve modifying cell membrane structure, interfering with neurotransmitter synthesis and release, and inhibiting enzyme activity. These mechanisms explain how drugs exert their effects at the cellular level and are crucial for understanding drug efficacy and safety. Proteins such as enzymes, ion channels, and carrier molecules are common targets for drug binding. Enzymes can be selective or non-selective, and their inhibition can be competitive or non-competitive. Ion channels and carrier molecules also play significant roles in drug action by facilitating or blocking the movement of ions and molecules across cell membranes. Ion channels are critical targets for drug action, as they regulate the flow of ions across cell membranes, affecting cellular activity. Drugs can modulate these channels to produce therapeutic effects. Carrier molecules, which transport substances across membranes, are also important drug targets. Receptors are specialized proteins that drugs bind to, initiating a cellular response. The concept of receptors was introduced in the late 19th century, and they are now known to be crucial for drug action. Receptors follow the law of mass action, where the drug's effect is related to its concentration and receptor occupancy. Receptors have specific properties, such as saturability and specificity, and can be classified into types like ionotropic, G-protein coupled, kinase-linked, and nuclear receptors. Each type has distinct mechanisms and roles in drug action, making them important targets for therapeutic intervention. Receptors propagate regulatory signals within cells, amplify these signals, and integrate various regulatory pathways to maintain homeostasis. Understanding receptor functions is essential for developing drugs that can modulate these pathways effectively and safely. Structure-activity relationship (SA) studies how a drug's chemical structure affects its interaction with receptors and its pharmacological properties. Small modifications in the drug molecule can lead to significant changes in its activity, making SA crucial for drug design and optimization. Affinity refers to a drug's tendency to bind to a receptor, while intrinsic activity measures the maximum effect a drug can produce. Partial agonists have less intrinsic activity than full agonists. Selectivity is the drug's ability to affect specific receptors or cell populations, which is important for minimizing side effects. Specificity is the ability of a drug to produce a particular effect by acting on a specific receptor type. Sensitivity refers to the lowest dose required to produce a given effect. Understanding these concepts helps in designing drugs that are both effective and safe, with minimal side effects. Drug tolerance can be natural or acquired, requiring higher doses for the same effect. Natural tolerance is seen in species like rabbits, while acquired tolerance develops from repeated use. Cross-tolerance occurs between related drugs, and tachyphylaxis is a rapid decrease in response to a drug. Drugs interact with receptors in various ways. Agonists activate receptors to produce a response, with full agonists eliciting the greatest response. Partial agonists activate receptors but with less intensity. Antagonists inhibit the action of agonists by binding to receptors without activating them. Competitive antagonists compete with agonists for the same receptor, while non-competitive antagonists bind elsewhere. Inverse agonists produce effects opposite to those of agonists by interacting with the same receptors. Agonist-antagonists act on multiple receptor types, producing mixed effects. This graph illustrates the varying levels of response elicited by full agonists, partial agonists, competitive antagonists, and inverse agonists as drug concentration increases. Chemical bonds between drugs and receptors vary in strength, from strong covalent bonds to weaker hydrogen and van der Waals bonds. The drug response is related to the dosage administered. The graded dose-response curve shows the relationship between drug dose and response intensity in a single biological unit. It highlights the concepts of threshold dose, ceiling dose, and hyperbolic curves. The quantile dose-response curve represents the response of a population to varying drug doses. It is used to estimate effective and lethal doses, and therapeutic indices.